We're very honoured to have a researcher who's emerging in the field give us an overview of the research they're doing in the development of treatments. Now, John Lee started his MND research career in 2008 as an honours student, working at the University of Queensland with Peter Noakes and Trent Woodruff. Now, John has actually continued this research and he's completed a PhD in 2014 where he specifically looked at the role of inflammation in motor neuron disease. And since then, he's actually gone on getting a fellowship from the MNDRA to conduct research looking at inflammation and metabolism, working between the clinic as well as the research lab with Professor Pamela McComb and Professor Trent Woodruff. And I'm looking forward to your talk discussing the role of PMX205 in clinical trials for motor neuron disease.
Thanks, Derek. So before I start, it is a privilege to have this opportunity very early in my career to actually present the work that I've actually done to the wider MND community. And also, before I proceed, I'd like to acknowledge MNDRA for giving us the funds to actually support the work that we're actually doing. So the work that our group is actually interested in is the immune system. So the immune system is the cells in our body, mainly in our blood, which protects and fights pathogens such as bacteria and viruses. So like when you catch a cold or when you actually get infections. So what this does is it actually initiates a key response to tissue injury and infection, and we actually call this inflammation. So what this does is it actually causes reddening and swelling as you have a rush of these blood cells going to these sites of injury and infections, as it is body's attempts at self-protections. So in addition to our immune cells in our blood, we also have these immune cells of the brain and spinal cord, and we actually call these microglia. So as you can see here in this picture, it looks like a star spindle shaped like in a normal brain and spinal cord. However, once we get these toxic stimulus or these injuries to our CNS, these microglia can go from this spindle star looking shape into a more circular shape, which is what we call microglia activation. And what this does, this actually can lead to the inflammation that I was talking to you in the previous slides. So what actually activates the immune system in MND? So on the top left here, we have a healthy motor neurons. And in MND, due to genetics or environmental factors, this can actually damage the motor neurons. And this damaged motor neurons can actually then lead to cell debris or the release of protein aggregates. And we all know that protein aggregates are like those pathological proteins that we all hear about, which is like the SOD1s, TDP43 and the C9orf72. And then the interesting thing about this is that this can actually lead to the activation of the immune system. So like I was saying, cell debris and aggregates from the damaged motor neurons can actually activate the microglia, the immune cells of the brain and spinal cord. And what this does, it can actually destruct a healthy motor neurons by releasing toxic molecules. And what this does is it can release cell debris and protein aggregates. So what this produces is like a cycle of inflammation-mediated neuronal death. So more of the neuronal death that you actually see, more neuronal death you're going to get. So what we want to do, and our approach, is to actually block the activation of these microglias and also the destruction of the motor neurons in the hope that we can actually halt the disease progression in MND. So one part of the inflammation that can do this is called the complement system. So this has been around for a very long time. So it's been discovered 100 years ago by the Jules Brodé. This is part of our innate immune system. So it's like our first defense against foreign pathogens and toxic stimulus. So what this is, it consists of cascade of proteins or targets. So it goes from C1 all the way to C9, which this can then lead to the bacteria death, which we don't want in our body. But what our group is actually interested in is this middle component here called C5. So what this does is that it can actually produce this target, C5a, which can then bind to its own, we can call it receptor, but for us we can call it a car. So it gets into its own car. The C5al1, which then can lead to inflammation, and this is what we find in MND. So to test this theory, what we did is we used an MND mouse model. So what we've done is we actually got a known mutation, such as the SOD1, and then introduced it into a mouse to produce a SOD1 transgenic mouse. So this is like a gold standard mouse model that we use in MND research, as this actually causes muscle weakness, body weight loss, and death due to respiratory failure, like what we see in MND patients. So initially what we've done, we looked at a normal mouse spinal cord. So on this picture on the left here is a normal mouse. So you can see that the green is like the immune cells that I was telling you about, and red is a complement activation. So what you can see here is that in a normal mouse spinal cord, we don't actually see any complement activation. However, when we look at an MND mouse spinal cord, you can see that there's a distinct difference between a normal wild-type spinal cord. So you can see, due to the motor neuron death that's occurring in the spinal cord, we see that the complement system itself is getting activated and it's actually getting switched on. So what we can actually conclude from this is that complement system itself is actually activated in MND spinal cords. So what our lab has done now is to actually develop a drug named PMX205. So this drug is actually known to block the complement system. And the good thing about this drug is that it can actually get into the brain and spinal cord, as one of the limitations of treating a CNS neurological disorders is to actually get the drug that we want into the brain and spinal cord. So what we've done is we've treated that SOD1 mouse, the MND mouse model, with our PMX205. So this is what Mary Louise and Mark were showing. So this is looking at the lifespan of these animals. So the orange is the placebo, so the mouse that's not actually getting the drug. So you can see that this is the proportion of these animals at endpoints. However, when we looked at the PMX205 treated animals, you can see that there was a shift in the actual lifespan, which tells us that if we block our target of interest, C5AR1, which is part of the complement system, then it can actually increase the lifespan of these MND mice. So following on from this, we also were interested in looking at the muscle strength. So in orange, you can see the placebo. So these mice can decline in muscle strength over time as the disease progresses. So the important thing about PMX205 is that you can see the blue line, it also declines, but however, in this time frame here, you can see that there's an improvement in the muscle strength of these mice. So in addition to the increase in the lifespan of these mice, we've also showed that treating these mice with PMX205 can actually improve muscle strength, which might be good for the MND patients from our lab. So this is a video of a placebo-treated mice. So normally a mouse tends to move a lot quicker than what you can see here. And you can also see that their gait is very wobbly, so that their gait is not very normal. And if you actually see it closely, that this animal here can actually see that it's dragging its hind limb when it's actually walking. And a natural characteristic of a mouse is to actually stand on its hind limbs to actually navigate the surrounding. However, this mouse is not able to do so. So on the right, you see a PMX205 treated animal. So you can see that the movement is a lot quicker than the one that you see on the left. And you do see that the gait is less wobbly compared to the one on the left. And you can also see that it's not actually dragging its hind limbs. And if we wait for it, it should be able to stand on its hind limb to actually navigate. Yes, there we go. So what this has told us that in addition to increasing the actual lifespan and the muscle strength that we're seeing, that by blocking our target of interest C5AR1, the complement system, it actually improves this pathology that we're seeing in these MND mice. So in addition to showing the effect in mouse models, what our lab was interested in was to actually show if this target of interest that we're interested in was also implicated or is increased in MND patients. So what we've done is we've looked at the C5a levels in the immune cells of MND patients. So as you can see on the left, that the MND patients' immune cells actually have higher levels of C5a compared to the control immune cells. And in addition to that, we also show that the C5AR1 is also increased in these immune cells of these MND patients compared to the controls. So in addition to what we showed in the mouse models, we also showed that the C5a and the C5AR1, which is our target of interest, is also elevated in these immune cells in MND patients. So where do we go from here? So what I've shown you today is that we're heavily doing this preclinical testings at the moment. So we've already shown, what I've shown today is that we've shown that this drug that we're working with, PMX205, actually has effect. So it has an efficacy in MND mouse models. So what we need to do is, because working with mouse is actually very small, we need to actually scale up the drugs, because when we're actually doing safety testings in other species like rats and dogs, we actually need quite a lot. This is actually ongoing at the moment, and we've actually completed the first scale up, which is about 300 grams of our drug, PMX205. And we've also done a safety trials in rats in China, and the actual results are very promising. So following on from this, what we hope to do in the late 2018 is to actually contact TGA and the FDA to get approval to see if we can actually proceed with the drug into human clinical trials, which then would go into phase 1 clinical trials where we're actually testing out the safety of the drugs in healthy participants in addition to MND patients. And once that's all approved, then we will proceed into phase 2 clinical trials where we're actually looking at the efficacy of the drug in small cohorts to begin with, where we were hoping to do this in Australia, and then we'll actually proceed that into a larger cohort, including USA. So today, I hope that I've shown you today that complement the system, C5AR1 particularly, activation contributes to the disease pathology in the MND mouse, and that this target is actually elevated in human MND patients. And I also showed you today that PMX205, which the drug that we have developed, actually blocks C5AR1 and ameliorates the MND and MND mouse models. So what this does is that PMX205 is ready to progress into the formal preclinical safety trials, is what we're doing at the moment in preparation for human trials. This work wouldn't have been done with our wonderful research team based at SBMS at University of Queensland. So my mentor, Trent Woodruff, Vinod Kumar and Raquel McGill. Our UK collaborators are Peter Noakes, Dr. Sharon No, Derek Stain and Susanna Mantovani, and our pharmaceutical company partner, Alcinex, CEO Alan Robertson, and my other mentors at Royal Brisbane Hospital MND Clinic, Professor Pamela McCombie and Dr. Robert Henderson. Thank you. Yes, mostly. So a lot of the times when we do these kind of experiments, you want to let it match your actual groups, like placebos and PMX205. So when we actually do a litter matched analysis, you always see that there's always an increase in lifespan and an increase in muscle strength in those ones that actually treated with PMX205.
Thank you, that's very promising. So thank you very much for joining us for this morning's session. I think we saw great examples of how the research is transitioning, not just from the beginning stage of understanding MND, but also how we can maybe modify this for the people living with MND. So thank you very much again for your attention. Thank you for the speakers this morning, and I hope to see you after our lunch. Thank you very much.